Very warm good morning to all of you. Welcome you all for the student interaction session. This opportunity has been given to the pharmacy department B from first year student. So we'll directly start with the student seminar. And for this, the whole class has been divided into 3 groups by Dr. S.C. Mahapatra and Dr. T.D. Dogra. They have visited our department on Monday, and this group was made randomly because in our classroom there were 3 rows. So out of these, 6 students are here to present, 3 will present and 3 will be the resource person for this presentation. So first presentation we will start with Miss Nikita, student of B Farm first year. So I call upon Miss Nikita.
And directors of different departments, my teachers and all my friends. Today I, Nikita, student of B Pharmacy first year, is going to present a seminar on the topic sterilization. As we are going through the student seminar in the previous semesters also, but it is not at that much broad level. So today we are going through the seminar with the help of presentation, and my topic is sterilization. In this sterilization, I will going to tell you the contents, that is introduction, in which I am going to tell you about that what is sterilization, different methods of sterilization, and at broad level I am going to explain you about dry heat sterilization. Sterilization is a process by which we kill or eliminate transmissible agents such as fungi, virus, bacteria from a surface, equipment, foods which we use in our daily life or culture medias. Basically, it is a process by which a surface or a medium is made free from all the microorganisms. Next one is why sterilization. Being a part of pharmaceutical industries, we basically deals with the dosage form. And in this, one of the most important is preparation of sterile dosage form. So sterilization is needed to prevent the contamination in sterile products, transmission of pathogenic microorganisms, decomposition of food and fruit products which we daily eat or use in our daily life, to prevent the contamination of unwanted microbes, contaminations in aseptic areas which are used for the preparation of sterile dosage form, and sterility testing which are used for the medications of patients. So the medications which are prepared should be sterile and free from all the microorganisms so that the patient should not suffer from any diseases. Next one is methods of sterilization. Sterilization is basically divided in 2 methods, that is physical methods and chemical methods. Physical method includes dry heat sterilization, moist heat sterilization, radiation sterilization and filtration methods. And chemical methods includes gaseous sterilization, for example formaldehyde, ethylene oxide, etc. and by using disinfectants such as creosotes, phenols, alcohols, aldehydes. Now in the physical methods, I am broadly going to explain you about the dry heat sterilization. As we all know that dry heat is the most reliable and rapid form of sterilization. Dry heat kills the bacteria by denaturation of bacterial protein, their oxidative damage, and by toxic effect of elevated levels of electrolytes. In dry heat, we kills the bacteria with the help of dry heat, which is widely used method. Dry heat consists of different methods, that is sunlight and drying, red heat, flaming, incineration and hot air oven. As we all know that sunlight consists of UV rays, that UV rays along with the heat rays responsible for the germicidal activity and kills the microorganisms. In red heat, we just put the material or the article which we are using on the flame until it becomes red hot. Examples are in the laboratories also we have used inoculating needles, inoculating loops which are used for the preparation of culture media like nutrient broth, nutrient agar, blood agar, glucose broth, etc. In flaming, we just pass the article over the flame without allowing it to become red hot. example like glass slide, so that it should not break and it should be sterile and free from all the microorganisms. Incineration is a process in which the material is reduced to ashes by burning them like animal carcasses. Their dead bodies are burned and converted into ashes. Examples are beddings, animal carcasses, etc. Now, hot air oven is one of the electrical device or an instrument which work on the principle of dry heat. The ideal temperature for hot air oven is 160 degree for 2 hours and 170 degree for 1 hour. We also use the temperature of 180 degree also for 30 minutes. In this process, we are having the hot air oven in which we can control the hot air oven with the help of the buttons which are present outside the oven. We can control the temperature and time. In this, we are having the double walled layer, which is bad conductor of heat from inside, and it is metallic from the outside. The aluminium trays are available on which the object is to be sterilized. Generally, glasswares are sterilized. We have also done in the laboratories also. We just wrap the glassware, test tubes, petri plates, etc. with the craft papers and just tie them with the threads and put it on the aluminium trays. Fan is also available so that the heat which is coming from inside should be uniformly distributed and glassware should not be affected and damaged by these. In hot air oven, we are also having the as we cannot sterilize surgical dressings or liquid media because it is dry heat is a poor conductor and is not heat level, and its penetration power is also low, so we can only sterilize glassware, petri plates and some of the talc, zinc oxide like products and pharmaceutical aids. Hot air oven is also is discovered by Louis Pasteur. It is one of the method of sterilization. Now my friends are going to explain the further methods. In the end, I just wanted to thank all the university officials to give me that opportunity and time. Thank you.
To our respected university officials, deans and directors of different faculties, my teachers and my dear friends. As my friend Nikita has already discussed about sterilization, need of sterilization, why sterilization is actually necessary for us, why control of microorganism is necessary mainly in pharmaceutical products, and different methods of sterilization. In broad, she also explains what is the method of dry heat sterilization. And taking a step ahead from it, I would like to introduce you from a different and the most effective and efficient method of sterilization known to be as moist heat sterilization. Basically, in this, I would like to introduce you from methods of moist heat sterilization, instruments used in moist heat sterilization, and the most important instrument, autoclave, its principle and its working. There are different methods of moist heat sterilization. Basically, there are three methods of moist heat sterilization. First one, sterilization at a temperature below 100 degrees Celsius, sterilization at a temperature of 100 degrees Celsius, and final one, which is the most effective and most efficient method of sterilization, sterilization at a temperature above than 100 degrees Celsius. Now, let's start with the sterilization at a temperature below 100 degrees Celsius. Sterilization at a temperature below 100 degrees Celsius can be takes place with the help of a process known to be as pasteurization, as it is given by a very famous Greek scientist, Louis Pasteur, who is also known to be as father of microbiology. This method includes mainly two sub-methods known to be as Holder method and Flash method. In this method, basically these methods are useful to sterilize those products which are reliable to heat or which can sterilize at a low temperature, that is less than 100 degrees Celsius. In Holder method, we heat or allow the materials which we want to make sterilize to get sterilized through the heat at a temperature of 63 degrees Celsius and allow it to heat for 30 minutes so that sterilization can take place easily, effectively and efficiently. And the second method of sterilization is Flash method. of flash method of pasteurization. In this, the material is exposed to the heat for 20 minutes at a temperature of 72 degrees Celsius, and it is beneficial in sterilization of those products which are used in household products. As you have daily seen in household products, at the packet of milk, it is written in bold letters that it is pasteurized milk. That is, it is used in the sterilization of dairy farm products like milk and butter. And after heating this, we are allowed to cool the material at a temperature of 13 degrees Celsius or less than that even, and this method is known as pasteurization. The second method of sterilization of moist heat sterilization is sterilization at a temperature of 100 degrees Celsius. This can be done with the help of two methods. First one, boiling, and second one is tyndallization. Boiling. We have already done, or we have usually done boiling daily in our household products, like we boil water to kill microorganisms, to make water sterile, to free from microorganisms, so that it can save, not harmful for us or safely for us. The same method applied here in pharmaceutical products, that boiling of materials, medias takes place so that these products are not harmful for us and are most beneficial for us. In boiling, generally the materials is exposed or boiled at a temperature of 100 degrees Celsius for 10 to 30 minutes, so that sterilization can take place easily, effectively and efficiently. And the second method of sterilization at a temperature of 100 degrees Celsius is tyndallization. It was given by a very famous scientist known as John Tyndall. The name is called to be tyndallization. In this method, the media or the glassware which we want to make sterilized is kept at a temperature of, boiled at a temperature of 100 degrees Celsius, mainly for 20 minutes successively for 2 to 3 days, like 20 minutes today, 20 minutes tomorrow, and 20 minutes day after tomorrow, so that sterilization rate can increases, and the material which we want to make sterilized can sterilize more effectively, efficiently. The third method of moist sterilization is sterilization at a temperature above than 100 degrees Celsius. It is the most effective method of sterilization which we have used in laboratory also to sterilize different medias like nutrient media, agar media, blood agar media, glucose medias. And sterilization at above 100 degrees Celsius can take place with the help of an instrument which is known to be as autoclave. We have used this autoclave in our laboratory also to sterilize medias, glasswares, and it is basically autoclave works on the principle of moist heat sterilization, and the principle is like that the greater the pressure inside the autoclave, the greater will be the temperature induced into the autoclave, which leads to the killing of or denaturation of bacterial cell wall, which leads to the sterilization of material which we want to make sterilized, which we want to make free from microorganisms, which we want to make not harmful for us and beneficial for us. Now I would like to introduce you with the instrument construction of autoclave. It is the outermost view of the autoclave. This is pressure regulating valve is present here, which regulates the pressure inside the autoclave. This pressure gauge, it tells about the amount of pressure inside the autoclave. The safety valve is present here to remove the excess of steam inside the autoclave. This autoclave lid is the outermost cover of the outermost part of the autoclave. A pair of handles was given also to carry the autoclave from one place to other. Autoclave body protects the insider part of the autoclave, and a steam releasing valve and a vacuum releasing valve are also present to release the excess of steam and vacuum. And outer stand given also to stand autoclave at any place in our laboratory. And it is an innermost view of the autoclave, so that you can understand how actually the autoclave works from inside. And autoclave from inside has a very chamber which is fully filled up of water, and a pair of coil is present which through electricity get charged and convert that water into the steam. So that steam sterilize the media and the equipments which we want to make sterilized in autoclave. Autoclave working. As I told you about instrument, its principle, its construction, now I would like to introduce you with how that instrument works. Basically, it is an instrument which is used to sterilize glasswares, that is the apparatus which we use in laboratory, and the different types of medias which we use in laboratory to perform our experiments. And basically in this heat in the form of saturated steam, that is conversion of water into the steam, is used in autoclave to kill the microorganism to make the product sterile. In this the temperature is set to be at 121 degrees Celsius at a pressure of 15 psi, that is per square inch, and it is allowed in the steam, and the materials is allowed in the steam for 15 minutes to get sterilized of nutrient media, and of different methods of sterilization can also there, but is the most effective and most efficient method of sterilization, as we have performed this experiment in our laboratory also.
Can I just interrupt? This 15 is 15 pounds per square inch goes. Yes, sir. Carry on, please.
At last, I would like to tell you about the basic advantages of autoclave also. As my friend Nikita has already discussed about hot air oven, but this autoclave, why should we call it as most effective and most efficient method of sterilization? There are some advantages of autoclave also over hot air oven. That's why we use this method. Like hot air oven can not kill those bacteria which bear spores, which can kill at a high temperature. But this autoclave can kill those bacteria, those microorganisms also, which can kill at a high temperature. The second advantage of autoclave is that it is most rapid method of sterilization. As I told you, it sterilizes the media glassware only in 20 minutes. That's why it is the most rapid and most effective method of sterilization. The sterilization in autoclave can be done in evenly if its penetration power is very high, and the material or the media or the glassware which we kept in the autoclave can be sterilized very, very, very effectively. At last, I would like to say and would like to end my presentation with a motivational quote, that is, setting goals is the first step to conversion or to turn the invisible into the visible. That is, it's never too late to done anything. Always try your best, gives your best, you take best from everyone. Thank you.
I am Hamid Abbas from B Pharmacy, first year, representing my presentation of the topic sterilization by filtration method. As my friend Keshav and Nikita discussed the topic sterilization and its different methods, I am continuing the topic sterilization by discussing the another method of sterilization, that is sterilization by filtration method. The content that I am showing you or discussing in these slides are sterilization by filtration method and different method of filtration. Sterilization by filtration. The method named sterilization by filtration is composed of two words, filtration and sterilization. Sterilization is killing or removal of any kind of microorganism from the pharmaceutical product, and filtration is the process by which we separate any solid particle from the solution. This is the non-thermal method of sterilization in which we use no heat, less economic, and not use any costly equipments in this method. For the sterilization of that product that are denatured by the heat and the steam, we sterilize by the method of sterilization by filtration. That products are eye drops, antibiotics, separation of bacteriophages, and isolation of microorganisms. General steps for the filtration. For any type of filtration or filtration process or filtration method, these steps are similar. Make a solution. If the mixture is present in the solid form, not present in the liquid form, then make the solution. Use any kind of filter for the filtration method and pass out the solution from the filter paper and collect the filtrate and the filter. Different method of filtration. On the basis of filters used, we classified the methods. Filtration on the basis of filtration by asbestos filter, filtration by sintered glass filter, filtration by filter candles, filtration by membrane filter. Then first filtration method is filtration by asbestos filter. This is In this method we use asbestos filter, that is magnesium, made up of magnesium trisilicate, and single this filter is single use and disposable after the process of filtration. In this method we take a conical flask, funnel and the asbestos filter. We attach the asbestos filter on the funnel and pass out the solution that we want to be filtered, collect the filtrate and the filtrant. This is used for the separation of this is used in the sewage plants for the separation of or for the purification of water, and for the in industries also, oil industries, for the separation of for the purification of oils. Next one is filtration by sintered glass. Sintered glass. These glass are made up of borosilicate, that is finely powdered and molded into a sheet. Molded into a sheet that is very costly and not used after sterilization. Used after its sterilization. In this method also we take a conical flask, funnel and attach a sintered glass disc on the funnel and pass out the solution that we want to be filtered, collect the filtrate and the filtrant. Filtration by filter candles. This is we use for the purification of water in our houses and in industries also. In this method we use a filter candle that have inside that have, this is a picture of industry filter candle which have many tubes inside it. In our houses filters we have only one tube inside it that is used for the filtration of soil and solid particles present in the water for after present in the water that is we want to be purified. And in this we use pressure for the filtration. Membrane filter. These filters are made up of cellulose and used after the sterilization into the autoclave, that is another physical method of sterilization. In this method, these filters have the pore size of 0.01 to 10 micron and used for the isolation of different kind of microorganisms and bacteria. In this method we take a conical flask, filter membrane filter and a clamp which is connecting the funnel, filter membrane and the conical flask, and we pass out the solution, select the filtrate and the filtrant. This filter membrane filter is sometimes used for the growing of any type of microorganism that we collected from the water that we solution that we pass out from this filter. Thank you.